So, hello everyone. So, good morning, good night, good evening. So actually, it's really my great honor to have this chance to present my talk here. So I would like to thank Rachel for her kind invitation for this wonderful BioEM talk. So I'm Faison from Institute of Biophysics, Chinese Academy of Sciences. So I have been in biological electron microscopy research field for 15 years. My major research field is structural biology and also biological imaging technology. And so I used cryo-EM to study the structure of micromolecular structures, and also use the BioEM method to study the architecture of cell and the tissue to address some biological question. So currently, I'm the leader of the Center for Biological Imaging in our institute. And today, I will give you a brief introduction about our biological imaging center and present some technology developments in our center from specimen preparation to imaging technology. So now a brief introduction about our biological imaging center. So our center is a kind of core facility, a kind of research platform by combining electron microscopy and fluorescence microscopy together, and we provide open services to our users, and the technology to try to image biological structure from nanoscale to mesoscale in 3D in nanometer resolution. And currently we have 16 engineers and 14 engineer assistants, and we also have a couple of chief scientists to help us to for their strategy and the future technology development. And for electron microscopes, we have a couple of transmission microscopes from low end to high end cryo-EM microscopes, as well as scanning electron microscopes. And we provide open services to our users in Beijing area, or also in China. And sometimes we also provide services to other countries. And our service includes molecular imaging, ultrastructure imaging, and molecular localization and multi-scale imaging. Besides besides imaging techniques, we also provide services for sample preparation. So as I know, for our BioEM talk, the major topics, one of the major topics on BioEM talk is related with sample preparation. So just as Renchi mentioned, so many of us countries are still following the mature protocols 30 years ago. Yeah, so actually this kind of work are also the routinely performed in our center. And we also provide the image analysis and the data processing services to our user. The one major services in our center is to provide the ultrastructure study of a cell and tissue sample, and the second direction, the second kind of service in our center actually is a cryo-EM. So now it's quite popular in the field. So we provide the services from sample preparation to data collection and structure determination to help our user to determine the 3D structure for their molecular complexes. And for the third kind of services, so we also provided the volume EM technology to our user. So it's kind of a series, continuous series sections, electron microscopy technology. So here there's some technology. I'm sorry. So there's some problem with it.
We are getting a feedback from.
Okay, is it fine?
Okay, it's fine now.
Okay.
Yeah.
Yeah.
Dave,
So
do you have BioTown open?
Yeah. Okay.
Maybe close the browser because we might have an echo.
Okay. Let me close the BioTown.
No.
Is it fine?
Yeah,
Hello?
now it's much better. Keep going.
Okay. Okay. No, that's great. Yeah. Okay. So this is another kind of services provided from our center. So it's kind of called a volume EM. And as a BioEM leading facility in China, so we have also the responsibility to organize serious workshops to introduce technologies to our user from sample preparation to imaging and to image processing. So in each year, we organized 1 or 2 workshops to our users. And the country, we have a large user groups, probably about 100 user groups. And many users can use the service of our biological center to have a very good production and publish many good papers. And in the following couple of slides, I would like to introduce some of specific and featured technologies in our center. The first one is about the cryo-EM data collection pipeline. So we successfully built a high throughput cryo-EM data collection pipeline for single particle analysis. And from sample preparation to grid screening and to high throughput data collection and real-time image processing and remote monitor. And the collection speed was improved gradually with some new technology developed. And as you can see here, so now we can achieve 300 micrographs per hour. It's a very high speed data collection. And now our users can only need to collect 1 day data and to achieve to solve the near atomic resolution of their micromolecular complexes. And the second is a kind of technology for grid production. And as we know, the grid is very important for electron microscopy. And we invented a couple of cryo-EM grid, including this Holy Array carbon grid and also the graphene grid. And recently, we developed a new cryo-EM grid. We call it amorphous nickel titanium alloy film to replace the carbon film. And using this metal film, we can achieve improved uniform distribution of particle and compared to the conventional carbon particle. And use this metal film, we can also achieve a better resolution for single party analysis. And for the third one, just as Rachel mentioned just now, so BioEM is still a very important technique to address biological question. And the major thing is about how to study the structure and ultrastructure complex system. And so cryo electron tomography is another technology to study the ultrastructure of the cell and the tissue. But how can we prepare a good sample is always challenging. So in recent years, cryo focused ion beam is a kind of emerging technology for preparing cryolamella for cell sample. And our center, a couple of years ago, our center developed our own cryo-fib technology. We call the D-cryo-fib. And we designed our own shutter and dual, and we achieved cryo-fib, cryolamella preparation for a couple of cell sample. And using our D-cryo-fib technology, so we can study the maturation process of marine herpes virus in the cell, and we can see the nuclear capsid maturation in the nuclear, and its primary amyloid variant formation in the cytosol, and its interaction with ER. And we also can visualize its secondary developmental process and see the mature virus in the vesicle. So this is the images from cryo prepared sections of a cell achieved by cryo-focused ion beam. And it's comparable to our conventional BioEM method, but using cryo-EM preparation method, we can see more structural details. For example, here we can see the tangementation protein complex between the membrane and the virus. And so cryo-focused ion beam has been proved quite successful to prepare cryolamella of cell sample. But how can we prepare cryolamella of tissue sample? So this is another technology we developed. We call the VHARD cryo-fib. We try to combine high pressure freezing and cryo ultramicrotone trimming and cryo-focused ion beam together to prepare cryolamella of tissue sample. So here is a movie just to show the whole workflow. And this workflow was just recently published in Journal of Structural Biology. So this is a tissue block. We use a vibrotome to prepare a semi-thin section of the tissue. Normally the thickness can be cut to 50 to 100 micrometer thickness. Then we put this tissue slice for high pressure freezing. So we designed a spatial carrier, a high pressure freezing carrier. We put the tissue into the carrier, and this is an auxiliary ring to protect the carrier. And this is a cryo protectant. And this is a sapphire disc for high pressure freezing. So after high pressure freezing, the tissue sample is frozen, is fixed. And after high pressure freezing, so we put the carrier into the cryo ultramicrotome and perform trimming procedure. we use a diamond knife to trim the metal material and make the cryo-fixed block exposed. And we make a thinner exposed tissue with a thickness about 20 micrometer. Then we transfer this pre-trimmed carrier into the auto grade. So this auto grade is used for high resolution cryo-electron tomography data collection in thermofacial Titan cryos. And then we put this auto grade into the shutter and transfer to cryo-focused ion beam machine. So this is an electron beam and then the ion beam. And we use a ion cryo-focused ion beam to further trim the tissue block and to prepare the cryo-lamella with a thickness about 200 nanometer. It's ready for cryo-EM imaging. And the last step, we transfer the prepared cryo-lamella into cryo-EM for data collection, especially for tomography data collection. So this is the whole procedure. And so we cut the tissue block and with this tilt angle 45 degree to allow we can collect the tilt series for 3D reconstruction. And for we can also achieve a large area of lamella. And for each lamella, we can collect the dozens of tomography data set. So it's a kind of high throughput way. And this is an example, some example we collected using our workflow. So the specimen is rat skeletal muscle. So this is a raw tilt imaging. We can clearly see the mitochondrial and the M line and the Z line and the actin filament. So after reconstruction, we can clearly see some connection. So this is a muscle sample. We can see the T-tube ultrastructure here and also the mitochondria. When we zoom in, we can clearly see there's some protein density between two membranes. So one is a plasma membrane, and another is called sarcoma membrane. So there's the retinine receptor protein complex is supposed located between these two membranes. And another sample we tested is a kind of rat liver tissue. So we can clearly see the mitochondria and the ribosome. And after subtotal averaging analysis, we can get the ribosome structure, in situ ribosome structure from rat liver tissue. And the structure is quite similar to the previous published single particle analysis. So this is our recent published workflow to prepare cryolamella of tissue sample. And another kind of technology in our center is related with correlative microscopy. And we developed the workflow for cryo-correlative fluorescence and electron microscopy. So we call it the HOPE cryoclaim. So we try to put our specimen in a vacuum. So here is just a movie to show the workflow. So we put the vitrified or cryolamella into a cryo holder, and this is the vacuum chamber we developed for cryofluorescence microscopy. Then we use this inverted fluorescence microscope to image our cryo sample. So there's some fluorescence beads located. After locating the interested region, the target region, so we just load the cryo holder into the cryo microscope. And after loading, then we can check our cryo sample, and by merging the EM micrograph with a fluorescence microscope, we can target we can identify our target region to see the ultrastructure. And another thing about in our technology in our center is a technology called series section volume electron microscopy, and the technology we developed is called AutoCAD SEM. So this is a video to show the workflow, and so this is the reason embedded tissue sample, and we just put it in a microtome to align the specimen with diamond knife. And this is an automatic collector we developed. We call it AutoCADs. And then we align the collector with a diamond knife and put the water in the diamond knife bolt. Then we started the automatic collection, section collection. So using this technology, we can collect series sections automatically. Normally, we can collect 10,000 sections in one day. And after collection, we just cut the tape onto the wafer and put the wafer into the scanning electron microscope for the subsequent imaging. So this is just one example of how we can collect the series SEM images of C. elegans, and then we can draw. We can make a segmentation of this neuron cell, and to see how these neuron cells are connected. And since our biological imaging center is combining electron microscopy and the fluorescence microscopy together, so in our center we also developed some advanced fluorescence microscopy, and so recently this new technology is called GI-SIM. It's called grazing incident structure illumination microscopy, developed by my colleague, Dr. Dong Li. And using this new super resolution fluorescence microscopy, the resolution can be improved greatly from the conventional fluorescence microscopy, 300 or 400 nanometer to about 100 nanometer. And using this for fluorescence microscopy, we can image the live cell, so we can see the dynamics of mitochondria with the different height and different shape. So this is just a brief introduction about our biological imaging center and what kind of feature and specific technology we can provide. And these technologies can enable our users to provide our users additional alternative approaches to address their own biological questions. And for the second part of my talk, I would like to report our recent technology development in collaboration with Thermo Fisher Scientific, led by Dr. Ivan Lezak. And our engineers, Dr. Xujing Li and Dr. Xiaojun Huang, are the major contributor to this work. And the technology we're trying to develop is called IDPC scanning transmission electron microscopy. Actually, this technology was invented and developed by Dr. Ivan Lizak, and it has been quite successfully proved. It has been proved that this technology is quite successful for material science study. And in this work, we try to explore whether this technology is also suitable and can also provide whether it can provide some unique feature for biological sample. Okay, so as we know, so our traditional transmission electron microscopy has been proved quite successful to study the ultrastructure for our of cell sample. We can see the many the cell organelles by transmission electron microscopy. However, besides TEM, transmission electron microscopy, there are still other kind of electron microscopy technique. One is called scanning electron microscopy, SEM, and the third one is called scanning transmission electron microscopy. And the SEM, as I just show you, so SEM has also been nowadays widely used for biological EM, for biological sample, and especially used for volume EM. So that's called, there are three kind of method. One is called focused ion beam SEM, and the second is called block phase scanning electron microscopy, and the third one is called series section SEM. And these three different techniques are very useful to study the 3D morphology and the 3D architecture of our complex tissue sample. And it has been proved quite useful in developmental cell and the brain science. And but how about scanning transmission electron microscopy? And so for scanning electron microscopy, it just use a focused electron beam on the surface sample, and the transmitted electrons are collected. But according to the electrons collected in different regions, they are STM can be classified into two kind of technique. One is called bright field stem, the BF stem or ABF stem. So it collects electrons within the convergence angle. So this is the bright. It can form a bright field imaging. And the second category is called dark field stem. So it collects electrons outside the convergence angle. So there are two kind of technology called ADF stem and the Hardiff stem. As we can see, for bright field stem, so we can get the image just like the conventional TEM. And about for dark field STM, so we can get inverted micrograph image compared to conventional TEM. And how about the resolution and the depth section of the STM? And so the resolution and the depth of focus is related with the convergence angle. And for the larger convergence angle, so the resolution will be increased. The size of probe is reduced, and the depth is also reduced. So theoretically, for a CS corrected microscope, so the probe can be smaller than 1 angstrom. So in material science, the scientists can use a CS corrected microscope to achieve atomic resolution. But for a CS non-corrected system, so there is one critical convergence angle. For example, this is a 200 kV electron microscope. So the critical convergence angle is about 7 milligram. And so we can manipulate the resolution and the depth of focus by changing the convergence angle. And scanning transmission electron microscope is actually has actually been applied for biological study, including the molecular mass measurement and to visualize the nanogold particles in immuno-EM. And also the bright field STEM and has also been successfully applied to study the 3D structure of biological sections. However, till now, the STEM technology has not been widely used in BioEM field. There are a couple of reasons. So the first thing is that for dark field STEM, its electron collection efficiency is quite low. It's several order magnitude smaller than in the bright field. So it's not suitable for those sensitive biological sample. And another thing about the dark field STEM is about the contrast. So its contrast is proportional to the square of elemental Z value. So that means for dark field STEM, so only the heavy atom is sensitive. So we cannot see the light elements. And how about the bright field? So the bright field, its contrast is non-linear, and its contrast transfer function also has a shortcoming about the oscillation, just as conventional transmission electron microscopy does. So there's no superior advantages compared to the conventional TEM. So that's the reason why STM technology is not widely used in biological application. But recently, there's another technology appeared, developed in some official scientific company. It's called IDPC STEM. So the IDPC STEM can generate linear contrast. So that's called phase contrast. And in material science, scientists has already proved IDPC STEM can be quite successful to image those sensitive specimen and also can image both heavy elements and the light elements, just as this slide show. So for the mental atom and also the organic atom can be visualized at the same time. So this is the advantages of IDP system. So in our study, we want to investigate whether we can also see some advantages of using IDP STEM for biological sections imaging. So we prepared two kind of biological sample using two different methods. One is a traditional sample preparation procedure, the osmium chemical fixation and the dehydration, EPON embedding, the ultrasonic section, and the post staining. And for the method 2, we just omit the post staining. So there's only one heavy atom staining procedure used in chemical fixation. So this is a kind of a minimum stained sections. So we used these two kind of specimen, these two kind of different protocols prepared specimen to compare RDPC STEM and the TEM. And the two kind of samples are used. One is a cell sample, another is a kind of tissue sample. And the first we want to study whether IDP STEM can have a better contrast compared to the conventional TEM. So to make a fair comparison, we have to make sure these two image methods must be controlled in the same illumination dose. So, for example, here, we try to a minimum condition, so the minimum dose, 1 electron per square angstrom. So for TEM, we merely can see the ultrastructure of the section. But when we increase the dose from 3 electron per square angstrom to 40 electron per angstrom, so as you can see, the contrast is gradually improved with the increased dose. So this is the TEM images. It's quite normal. But how about ITP system? So here, this is a ITP system. As you can see, even at the minimum dose condition, ITP system can still yield a fair contrast. We can see the double membrane ultrastructure of mitochondria. We also can see some protein particles outside. And when we increase the dose, the contrast is improved as expected. And then we compare TEM micrograph and ITP system micrograph by zooming in. So this is the zoom in the mitochondria. So we can see the TEM micrograph, and from the TEM micrograph we can see the membrane structure and the crystal of mitochondria, but we could not see some protein on the outer membrane from in the TEM imaging. But how about the ITP system imaging? And as you can see, the contrast is greatly improved, and we can also see some membrane protein complex located on the outer membrane of mitochondria. We can also see a lot of structural details inside the mitochondria. When we zoom in further, actually, we can see here, I docked the F1 ATP synthesis structure into this micrograph. It can be fairly fitted, so we can exactly see the ATP synthesis dimerized structure in ITPC micrograph. And when we can zoom in another region, here is a contact site between the ER-like structure and the mitochondria. So this is a TEM imaging. And then how about the ITPC imaging? So you can clearly see some membrane protein complex on the membrane. And then when we zoom in again, so there are actually two membrane protein complex outside the membrane, but we cannot see such structure details in TEM micrograph. So from this comparison, we can conclude so ITPC stem can yield a better contrast and also a good resolution in molecular details. Then we made a quantitative comparison by using this line profile method, and to measure the contrast of the different images. As we can see here, the ITP system can always have a better contrast than TEM micrograph. So this is the first kind of test. And how about the second test? So we want to test so whether ITP system can still yield a good image for the section with minimum staining. And for TEM, as you can see, for the normal staining and the minimum staining, for the minimum staining, the contrast is low as we expected, So when we zoom in, and we can see some ultrastructure here. So this is a mitochondrial double membrane, and this is some ER-like organelle. And again, so this for the minimum standard section, when we zoom in, actually we cannot see the ultrastructure clearly from TEM graph. But how about RTP stem? So as we can see, for the minimum staining condition, RTP stem can still yield a very good contrast. And when we zoom in, we can make a clear comparison. And for the minimum staining condition, we can in RTP stem we can clearly see there are two vesicle-like ultrastructure here, but we could not see in TEM images. So from this kind of experiment, we can conclude. So RTP stem can also be suitable to study the biological sections with minimum staining. I think this is very useful in the future, but because normally post-staining actually is not sometimes so we can avoid some hazard reagents.
Can I interrupt? We are running really late. I still have two more speakers. Sorry, because otherwise the webinar is going to cut us off.
Okay, so I will speed up.
Yeah, can we move your part of your talk? We can still present it during the live chat, but webinar here because we are limited with time. Yeah. Okay. So just I will finish soon. Okay. Okay. So this is the third experiment is about the thicker section. So RTP stem can also give a good contrast for thicker sample. So this is just some explanation why RTP stem is suitable for thicker sample, and we can have more discussion later. And so this is a depth section. So it's here just a live image, a movie to show we can just scanning the section from top surface to the bottom surface and to try to get the 3D structure of the biological section. And here just some example to here. So this slide just show the theoretical analysis why RTP stem is is better than conventional TEM. Then we can discuss later. And this is the conclusion. And in our study, we investigated the RTP stem, the potential application of RTP stem in biological application. And we proved the RTPC can result a better contrast at the same dose and resolve more biological ultrastructure in molecular resolution. And it also suitable for biological section with a minimum staining condition. And also it can has a unique advantage to image thicker sections without loss of contrast. Okay, so that's just the end of my talk. Thank you very much for your attention.